hello and welcome everybody to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we're happy you're making the time to dial in today. We're here with Dan Nomura today, and we'll be talking about reimagining druggability using chemoproteomics platforms. And now, without further ado, I want to introduce our guest, Dan. So Dan Nomura doesn't need much introduction, of course, but he is a professor of chemical biology and molecular therapeutics at the University of Berkeley in California. He's also an adjunct professor in the Department of Pharmaceutical Chemistry at UCSF. He is co-director of the Molecular Therapeutics Initiative at UC Berkeley, and since 2017, he has also been the director of the Novartis Berkeley Translational Chemical Biology Institute, focused on using chemoproteomics platforms to tackle the undruggable proteome. Dan is the co-founder of Frontier Medicines, a biotech company focused on using chemoproteomics and machine learning approaches to tackle the undruggable proteome. He is also the co-founder of Zenith Therapeutics, focused on targeted protein degradation of undruggable targets. Dan is on the scientific advisory boards of Frontier Medicines, Zenith, Deciphera, and many other biotech companies. And Dan is also a scientific advisor on the scientific advisory committees of the Mark Foundation for Cancer Research and the American Association for Cancer Research as well. He's an investment advisory partner at A16Z Bio and Health, an investment advisory board member at Droya Ventures, and an I partner with the Column Group. In 2025, Dan also became the editor-in-chief for Molecular Cancer Therapeutics, and academically, he earned his bachelor's degree in molecular and cell biology in 2003 and a PhD in molecular toxicology in 2008 at UC Berkeley with Professor John Casaida, and was a postdoctoral fellow at Scripps Research with Ben Cravat before returning to Berkeley as a faculty member in 2011. Among his honors are the National Cancer Institute Outstanding Investigator Award. He's also a Searle Scholar, and he received the Mark Foundation for Cancer Research Aspire Award. Dan, really honored to have you today. What an impressive vita you have. So really honored to have you today.
Thank you so much. And thanks for having me. I'm a big fan of Drug Hunter. So yeah, thank you.
That's amazing. That's amazing. Yeah, and we want to get to know you a little bit better. In the good old Drug Hunter tradition, we're starting with some warm-up questions for the speaker. So could you maybe talk a little bit about what inspired you to start a career in drug discovery?
Yeah, I mean, I was always interested in human health and biology growing up. And when I came into UC Berkeley as a freshman in college, influenced by my parents, they really wanted me to go to medical school, and so I was pre-med like everyone else out there. And I quickly realized that it's not necessarily medical doctors that are discovering new therapeutics and drugs. It's actually the biologists and chemists that are really pioneering discoveries into new targets and new technologies and new drugs that are curing the patients. And I really wanted to be in that space of creating new medicines that would be transformative for diseases. And I thought that would have a much bigger impact. And I remember in my freshman year, I was reading these papers coming out of back then, Ben Cravat had just started his lab at Scripps. This was back in 1999, and I read kind of his first papers on activity-based protein profiling, and that really blew my mind and opened up a whole area of science that I hadn't appreciated before. That most proteins in the human body had not been drugged yet, and in fact most proteins in the human proteome were really undruggable. And so I was really enamored all the way back then, back when I was an 18-year-old, and I just pursued that track my entire career.
Amazing. Yeah, I think it's incredibly inspiring for everybody to solve all those unsolved problems, right? So it's really great that even that early you got into that. Very inspiring. Dan, what's your favorite molecule and why?
I think I'm going to be a little bit biased here. I'm probably quite conflicted in saying this, but I'm very, very enthusiastic about my company, Frontier Medicine's pipeline of molecules that they have, certainly in the forefront with their KRAS G12C GTP and GDP state molecule, where it's really, really equal potent for both the on and off state of KRAS G12C, and I think that's been really differentiating in the sense that it's able to really overcome kind of the resistance mechanisms that have been observed with kind of the first generation KRAS G12C GDP state inhibitors. So really, really excited about the clinical data that's hopefully going to come out soon on that. And then, their whole remaining pipeline, including the P53Y220C covalent activators that show really, really remarkable efficacy there in those mutant patients and or mutant cancer cells. And also their KRAS G12D inhibitor that's coming out soon, and also their PI3 kinase RAS covalent disruptor. So really, really excited about that and how covalent chemoproteomic approaches have really allowed them to accelerate the drug discovery process and really come out with molecules that are exceptionally selective, right? And in addition to being potent, and I think that's really important in helping to mitigate off-target toxicological liabilities of these types of molecules going forward. So really excited about those molecules.
Amazing, amazing. Yeah, and I think it's kind of changing the paradigm. Like with covalency, there was a perceived safety risk traditionally, but I think those molecules show that that can certainly be overcome. So really, very, very excited about those as well. Can you talk a little bit about what you're most excited about in the drug discovery world today?
Yeah, I think I'm really just excited about the explosion of new therapeutic modalities in drug discovery these days. And to me, right, whether it's PROTACs or molecular glue degraders or covalent drug discovery approaches or DNA encoded library technologies, or in the biologic space, multi-specifics or cell therapies, or gene editing technologies that are coming out. These are all basically modalities that allow us bigger and more expansive access into the undruggable proteome. And so, this whole concept of 80 to 85% of the proteome being undruggable, with all of these new modalities now, I feel like we now have the full arsenal of capabilities, or we're starting to have the full arsenal of capabilities of being able to at least have the potential to tackle 100% of the proteome. Now, obviously, we're not there yet, but I see that that's now a possibility. Whereas, when I first started off in this field, it seemed really, really far away, right? And now it seems like it's within reach, and so that's incredibly exciting to me.
I 100% agree with you. No, that's super exciting that we now have all the tools in the toolbox to maybe go after these traditionally challenging targets. And love to hear more about this, of course, from you today. What would you tell your younger self if you could go back in your career? What would you give yourself as a career advice? Maybe also for the younger folks in the audience today dialing in. What is some advice you would be giving them?
Yeah, I think this is something I encourage everyone from undergraduates to graduate students to postdocs to junior faculty to kind of think about. And it's something that maybe I didn't think about early on in my career when I was starting off as an assistant professor. I remember thinking, oh man, there's this big bottleneck of tenure, and you have to get tenure and you have to achieve a lot of things before tenure, and at Berkeley that's only 5 years to get there. And so I was thinking always in terms of that kind of 5 year timeline of what I can achieve. But having passed that hallmark many years ago, the things I think about now are more on the 20, 30 year horizon of what we need to accomplish and what are the incremental steps to get there. And so um and I wish I was thinking that way, from the start, right? What are the big goals that we have to accomplish over 20 to 30 years, and what incremental advances can we make now to get there? So yeah, that's the advice I'd give to myself.
Awesome, awesome. Now, I think I read a study once that says we vastly underestimate what we can achieve in the long run, and we vastly overestimate what we can achieve in the short run. So I
I think
think
that's true. Yeah.
so. This is I think really great advice, Dan. All right. Lastly, to wrap this up, what is your favorite thing about Drug Hunter? What do you like most about Drug Hunter?
Yeah, I love so I'm now a subscriber of Drug Hunter, and I love that I can just go into a particular target that I care about and type it in, and I can get kind of all the background around that target, why it's important, what drugs or chemical matter might be out there already. And I think another really valuable thing about Drug Hunter is that you guys really dive deep into a lot of patent literature and really fish out like what the best molecule might be within that patent literature, and it just saves us a lot of time of having to do that ourselves, and so that's been really useful for us. So thank you.
Awesome. Yeah, amazing. Really appreciate that. Okay, so before we get started, I will show very briefly to the audience the Drug Hunter platform. So sharing my screen here now, and I wanted to show you the structure search today. So you can quickly go here on the top left and get into the structure search, and you can draw with your keyboard. For example, here I'm using ones and twos to draw an acrylamide. So we can search for an acrylamide. And what I want to do is we have three different databases. We have the FDA approved drugs, so we can quickly see what FDA approved drugs contain acrylamides. That search, just takes a few seconds, and we can get a list of compounds that contain acrylamides with their targets. When we click here, we directly go to the FDA page and can learn more about the access documents that come with these drugs. So it's very simple to learn more about those drugs very quickly. And then if you are interested in more in clinical candidates, so something that's, maybe right before a drug gets approved, what are some compounds that are in clinical development? How do they potentially differentiate from drugs? You can do that here in our clinical compound database. Again, you can sort them by year. So if you wanted to see, for example, the most recent ones, we can see those here. And you can kind of see how they're different or similar to the FDA approved drugs. You can see what are some of those compounds that are, kind of on the verge of potentially becoming FDA approved drugs. And if you even want to go one step further, you can go here to the Drug Hunter molecules. Again, notice that when you click back, your drawing is always be saved with all the molecules that you draw. So we can go structure search here in the Drug Hunter molecules, and that finds you kind of the most modern compounds here. You can sort that again by date of the article or by name, by target. You can go through that as well and see where they have been developments. And if you are interested in more details on any of those molecules, it's very easy to just click here, stay in the platform. And for those molecules, we would have case studies for you available. So you can learn more about the entire program, about the biological background, about why they used the specific warhead, how they optimized the entire compound. So very quickly, within a few minutes, you can get quickly started on a research project and save a lot of time for yourself. And with that, I will hand it off to Dan. Very excited to hear your presentation today on chemoproteomics platforms and thinking about how we can address the undruggable space. So thank you, Dan.
All right. So again, thank you so much for having me today, and I'll talk about some of our latest work coming out of our lab. So as Dennis alluded to, our lab is really interested in tackling the undruggable proteome, which I think is still the biggest bottleneck we face in drug discovery, that over 90% of proteins are considered undruggable because they don't really possess well-defined binding pockets that a small molecule can bind to affect their function. And undruggable proteins include things like protein complexes or intrinsically disordered and unstructured proteins, where it's really unclear where within these smooth interfaces or unstructured messes like this you can identify a binding pocket they can pharmacologically interrogate. And I would say that among the classes of undruggable targets out there, the least druggable class of proteins out there right now are probably these transcription factors, which are involved in protein interactions and protein DNA interactions. And they also tend to have high regions of unstructured messes. And so these, and there are many of these transcription factors that are very, very important in disease, such as, for example, in cancer with proteins like MYC and beta-catenin, or truncation variants of the androgen receptor like ARB7 or NRF2, or, for example, in inflammation with targets like IRF5. And these 5 proteins just represent the tip of the iceberg of tens of thousands of proteins that remained really, really intractable. And so our premise has been that if we can identify binding pockets, or what we call ligandable hotspots, across all proteins in the proteome, that would really enable us to effectively go after drugging the entirety of the proteome. And so to do that, we've been utilizing chemical proteomics and coupling that with covalent chemistry to both discover ligandable sites and ligand those sites directly in complex biological systems. We've also been using these approaches to expand the scope of targeted protein degradation technologies. And we've also been using these to go beyond degradation to enable new induced proximity-based therapeutic approaches, all towards discovering new therapeutics and modalities to really overcome this challenge of undruggability. And so having come out of Ben Kravat's lab many years ago, the underlying approach that we use is an approach that he pioneered called activity-based protein profiling, or ABPP, that uses reactivity-based chemical probes to profile proteome-wide reactive functional ligandable sites directly in complex biological systems. And these probes consist of broad kind of chemoselective electrophilic warheads, as well as these alkyne bioorthogonal handles that you can use in complex proteomes to engage aberrantly reactive nucleophilic amino acid hotspots in proteins. And you can subsequently click on some kind of analytical handle, like an enrichment handle, and then pull out your proteins and your modified peptides to shoot them on the mass spec to identify the sites of probe labeling. And these sites of probe labeling now constitute potential ligandable sites that can now be pharmacologically interrogated with covalent small molecules. And in aggregate, taking thousands of these data sets together, we've now identified over 100,000 potential ligandable sites across really the vast majority of the human proteome. And these now not only encompass active sites, but also allosteric sites, cryptic sites, sites within intrinsically disordered regions, post-translational modification sites, protein-protein interaction sites, and more. Now, to be able to more selectively interrogate these sites with more elaborated chemical matter, we've also built out large libraries of molecules that bear electrophilic warheads with perhaps more tempered reactivity that it can use to now screen against either recombinant protein to identify selective chemical binders against those targets of interest, or we can deploy these directly in complex biological systems with the chemoproteomic approaches, where you can envision treating your covalent molecule in vivo in humans or mice or living cells, and the ligands will irreversibly engage whatever targets it's going to bind to. So when you chase with your reactivity-based probes, the probe can no longer bind to those sites. So it's a simple competitive assay that you read subsequently with quantitative proteomics. And through this, you can subsequently identify the sites for your small molecule ligand displaced probe labeling, allowing you to identify exactly which amino acid and which protein target your ligand bound to, and perhaps more importantly, how selectively it bound to that target on a proteome-wide scale. So you can use this to enable, for example, target identification of a molecule that came out of a phenotypic screen, or you can use this to assess target engagement of your molecule against your favorite target of interest. And most importantly, again, you can assess selectivity, so you can couple that with medicinal chemistry approaches to not only assess or optimize potency, but also protein-wide selectivity of your molecule to reduce off-target toxicological liabilities. All right, so I wanted to talk originally about how we've used these kinds of approaches to enable targeted protein degradation strategies. So many of you know already there are 2 major flavors of targeted protein degradation. One is with these proteolysis targeting chimeras, or PROTACs, that were pioneered by Craig Cruz and Ray Shea, that are very modular in design, with these heterobifunctional molecules consisting of a protein targeting ligand linked to an E3 ligase recruiter to induce the proximity of an E3 to ubiquitinate and degrade your target. But there's still major challenges with this. If you have an undruggable target, finding your ligand is still challenging. And while there are over 600 E3 ligases out there, we primarily only have recruiters against 2 of these, against cereblon with the thalidomide and cyclic imide class of molecules, and another one against VHL. Uh, and that's because E3 ligases are also among the less tractable classes of proteins, although there's been a lot of efforts in trying to expand the scope of new E3 ligase recruiters. So these are still major challenges in the field of PROTACs. Now, there are also molecular glue degraders that are smaller molecular weight and perhaps more drug-like and able to access less druggable targets by accessing the shallow interfaces between an E3 ligase and a neosubstrate protein to glue those together to ubiquitinate and degrade them. But so far, most molecular glue degraders have been accidentally discovered or found through phenotypic screens. And it's been challenging to develop rational design strategies for molecular glue degraders in a target-centric manner. And so these represent still big challenges in the field of realizing the full potential of targeted protein degradation technologies. And so this is an area that we've been in for quite some time now, where we've been, for example, using chemoproteomic approaches to expand upon the scope of new E3 ligase recruiters. This is something we originally stumbled on with investigating the mode of action of this electrophilic natural product, nimbolide, where we found that this covalently reacted with a cysteine on an E3 ligase, RNF114, and in collaboration with Tom Moni's group, we showed that we could use this for PROTAC applications. We subsequently developed a covalent RNF4 recruiter that we used in PROTACs. In collaboration with Michael Rappa's group at UC Berkeley, we also developed a covalent recruiter against this reductive stress E3 ligase, FEM1B, and showed that we could use it in PROTAC applications. We subsequently went after core components of the ubiquitin proteasome machinery that are essential for cell viability to potentially, overcome potential resistance mechanisms that you might get from exploiting non-essential E3s like cereblon. And we were able to develop a covalent allosteric non-functional recruiter against an E2 ubiquitin conjugating enzyme, and again, use that for PROTAC applications. And we've subsequently also gone after developing covalent recruiters against cullen adapter proteins, again, essential for cell viability against col4 adapter proteins like DDB1 and col1 adapter proteins like SCIP1, and again, developed covalent recruiters against those as well. As relates to the rational design of molecular glue degraders, we've also somewhat cracked this problem by taking more of a chemistry-centric approach, where we've been starting with protein targeting ligands against well-established targets and then elaborating off of the exit vectors with a variety of electrophilic chemical handles to identify permissive covalent degradative handles, and in combination with chemoproteomics, permissive E3 ligase pairs that we can then subsequently transplant across a variety of protein targeting ligands to induce the degradation of those targets. And so a couple of years ago now, we were able to discover this covalent fumarate-based handle that targets an E3 ligase on this quality control E3 ligase called RNF126, which we were able to show was highly transplantable across a variety of different protein targeting ligands to induce the degradation of those targets. We subsequently then discovered an even smaller molecular weight vinyl sulfonylurea handle that targeted a unique cysteine on DCAF16 that, again, was highly transplantable across a variety of protein targeting ligands to induce the degradation of their targets. And we've subsequently now been able to develop or identify potential ligandable sites across the vast majority of the over 600 components of the ubiquitin proteasome system. Most recently, we were able to show that we discovered yet another permissive covalent degradative handle with the fumaramide handle that targets the set of unique cysteines, cysteine 173 and cysteine 178, on yet again DCAF16, which has turned out to be quite a popular E3 ligase, especially for covalent degraders. This is some E3 ligase that originally Ben Cravat and Xiaoyu Zhang discovered as a handle for covalent PROTACs, and subsequently Nathaniel Gray and Eric Fisher and Garrett Winter and many others have also discovered DCAF16 as the E3 ligase responsible for the mechanism of action of other molecular glue degraders. But this was a handle that seemingly was able to access a unique cysteine 173 and 178 within DCAF16, again, that seemed to be highly transplantable to enable the degradation of many different neosubstrate targets. And so it seems like this E3 ligase has many cysteines right at its substrate interface for some reason, to enable neosubstrate interactions that can be exploited with covalent chemistry and covalent degraders. And so that's kind of our latest efforts in this endeavor of trying to find permissive degradative handle and permissive E3 ligase pairs for the rational design of molecular glue degraders. But I wanted to spend some time on these undruggable targets that I've been talking about, right? So there are many targets that have been attributed to driving cancer and could all be therapeutic targets for cancer, yet only a small fraction of them have small molecule chemical matter associated with them. And that's because most of these targets are among the undruggable class of proteins. But if you mine our ligandability database, we've identified ligandable sites across the vast majority of many of these cancer drivers. So we can now systematically start going after enabling ligand discovery against many of these. So what about this transcription factor, c-Myc, that has eluded drug discovery efforts for many, many years? This is a very important oncogenic transcription factor that's amplified across the vast majority of human cancers and is thought to be a major driver of many human cancers. Yet it's been very elusive because this protein is not an enzyme, doesn't have an active site, is engaged in many protein-protein interactions, and it's also highly intrinsically disordered. This is an alpha-fold structure where it's mostly a stringy spaghetti monster, right? And so typically targeting intrinsically disordered proteins has been considered very, very challenging. Whoops, keep getting calls. When we mine our ligandability database in MYC, we find that both cysteine 186 and cysteine 203 appear to be aberrantly reactive cysteines within intrinsically disordered regions of MYC. Now, usually, again, targeting intrinsically disordered regions in proteins is considered impossible, but we conjectured that there must be some kind of perhaps transient structure that's being recognized that's conferring hyperreactivity to these particular cysteines. And so we thought maybe we could target those with covalent chemistry. Now, several years ago now, we found this covalent molecule EN4 that selectively engages one of these intrinsically disordered cysteines, cysteine 186. And through making alkaline functionalized probes, we were able to validate that interaction with both recombinant protein as well as show that we can engage MYC in cells with this covalent molecule. And in fact, this molecule is in fact functional. We could disrupt MYC-Max binding to DNA, and we could also disrupt MYC transcription activity in cells. And if you mutate that cysteine 186 to a tyrosine, you can attenuate that response. And when we looked at thermal stability of MYC in cells, we found that this molecule, instead of stabilizing the stability of MYC, actually caused destabilization of MYC. And that actually led to the ubiquitin proteasome-mediated degradation of MYC. And so you could see that MYC protein levels are lost, and you can rescue that with a proteasome inhibitor. So this wasn't just a MYC inhibitor. This was actually a MYC degrader. And that led to profound downregulation of a lot of MYC target genes and also anti-tumorigenic effects in vivo in breast cancer models that were driven by MYC. And so this was not your typical molecular glue degrader or PROTAC. This was covalently targeting an already kind of thermodynamically unstable region of the protein and causing it to further become destabilized and then get ubiquitinated and degraded. So we wondered, could this be a potential vulnerability within transcription factors that we can exploit? And so we went into our ligandability database and we're wondering how many of the sites that we've identified are within disordered regions. And perhaps not surprisingly, the vast majority of sites we've identified are within ordered regions, and that includes things like catalytic cysteines. But a good 12% of the sites we've identified, over 14,000 sites, are aberrantly reactive cysteines within intrinsically disordered regions of those proteins. Now, if you look at the target list within those sites, there's a whole bunch of transcription factors that have been shown to be very critical in disease, yet have remained elusive to kind of direct drug discovery efforts. And so we thought maybe this is a critical vulnerability that we could potentially exploit, and that these intrinsically disordered regions could be potentially ligandable sites. Now, one of the challenges, though, with that original EN4 molecule were that we weren't really clear on whether that molecule was just reacting with that cysteine in a purely reactivity-driven manner, or whether there was some kind of transient binding pocket that was being molecularly recognized and then subsequently forming a covalent bond to destabilize and degrade MYC. And that's a pretty big distinction, because if it was purely just reacting with that intrinsically disordered cysteine in a reactivity-driven manner, it's going to be pretty hard to get SAR on that molecule and to be able to drive down potency through improving affinity. And probably any potency gain that you get is just going to be reactivity driven and just increasing promiscuity of that molecule. And so we wanted to address that in a kind of chemistry-centric manner, because we're not going to be able to really ever solve the structure of an intrinsically disordered protein like MYC. And so in collaboration with Tom Maimoni's group, and this was led by a talented graduate student, Hannah Rosen, in my lab, and a talented chemistry graduate student in Tom Maimoni's lab, Kelvin Lee, where we really went back to the drawing board on warhead design, and we really tried to design warheads that were very tempered in their reactivity. And we settled on this sulfonyluresidine warhead that has quite tempered reactivity with things like glutathione and cysteine basally. And we also engineered stereochemistry directly into the warhead onto this kind of oxindole core that is highly diversifiable. And the idea here was, could we stereoselectively engage an intrinsically disordered cysteine on MYC to destabilize and degrade it? Because if we're able to do that, then that would really indicate that there must have been some kind of transient structure that could be stereoselectively recognized in MYC to subsequently covalently bind to it and destabilize it. And that would give us a little bit more confidence that, there must be some kind of transient structure that's being formed that we can potentially interrogate, and also to drive down potency. And so we made a library of these molecules and screened them for MYC degradation, and we were able to, in fact, find active hit, where we also found basically almost inactive diastereomer here. So this 236 molecule was able to dose responsibly degrade MYC, whereas this 419 molecule is essentially inactive and a diastereomer of this active molecule. And you can also see that by Western blotting that MYC protein levels are taken out, and you can rescue that with the proteasome inhibitor, whereas the inactive molecule, it doesn't degrade MYC. Now, you might think, though, that perhaps some of these stereoisomers might just have difference in reactivity basally. And so we made all the enantiomers and diastereomers of this active molecule. And when you throw it into a naked artificial peptide here, this was all done in collaboration with our close collaborator, Novartis. This is an artificial peptide with representative amino acids. First of all, we didn't know whether this was cysteine chemoselective. It is in this artificial peptide system, but there's really no difference in reactivity between any of the enantiomers and diastereomers here. So basally, there's no difference in reactivity. So the stereoselectivity that we're observing in degradation must be because we're recognizing some kind of protein environment. Now, in cellular settings, though, MYC engages in a diversity of different protein-protein interactions. So you might think that maybe we're creating a protein environment through a diversity of different protein interactions that MYC is engaging in. But what was really remarkable was that Novartis was actually able to generate full-length human MYC-max protein that was functional. And even in this recombinant protein setting, where we just have MYC and MAX and PBS in the test tube, we were able to show that this 236 molecule could stereoselectively engage MYC over the less active diastereomer. So even in this pure protein setting, we're seeing that stereoselectivity. We could also see that engagement of MYC in a stereoselective manner in cells as well, since these have alkyne probes on them, handles on them. We can throw them into cells and then click on a biotin handle and enrich MYC, and we could show stereoselective enrichment there in cells. We could also use this in chemoproteomic pull-down experiments and show robust engagement of MYC, although this original molecule does have over 100 off-targets by chemoproteomics experiments, so it's not quite selective, and I'll address that later on here. And when we compare the chemoproteomic profiles between the active molecule versus the less active diastereomer, we see also stereoselective engagement of MYC, although again, we see a number of off-targets here as well. When we do a thermal shift analysis on this, we show quite robust destabilization of MYC thermal stability with almost a 6-degree TM shift here. So this is yet again one of these destabilizing degraders. When we mapped its site of reactivity, though, we found now that this didn't react with that original cysteine 186, but now reacted with the other predicted abnormally reactive cysteine, cysteine 203. And what we were quite interested in was that it seemed like when we looked at the cytomodification of this molecule, not just with MYC, but also with many of its off-targets, we found that there was an enrichment in acidic residues 2 positions over from the modified cysteine. In this case, it was an aspartic acid 205 within MYC. And so we thought perhaps this acidic residue is either activating the cysteine or maybe even activating the sulfonyluresidine, or maybe in the absence of the cysteine, this molecule can somewhat reversibly interact with that aspartic acid. And consistent with that, when you mutate the cysteine alone, you still get degradation of MYC. When you mutate the aspartic acid alone, you still get degradation of MYC. But when you mutate both the cysteine and the aspartic acid, you now get full degradation, a full rescue of MYC degradation, indicating that it actually requires both of these residues to coordinate the reactivity of this molecule with MYC. Now, is this molecule functional? It is. You could see nice dose-responsive inhibition of MYC transcriptional activity in cells with the active molecule, but not the inactive diastereomer. When we do RNA-seq, we see a lot of genes changing, but when you do pathway analysis on these genes, MYC is the major pathway that's dysregulated. So it's quite selective in just manipulating MYC transcriptional activity. But one of the challenges with transcription factors is that not only are they difficult to target, but especially transcription factors like MYC, they also have very fast turnover rates. So they're constantly being synthesized and constantly being degraded. And that's a big challenge. And you can actually see that in this pancreatic cancer cell that has very fast MYC turnover. This 236 molecule shows loss of MYC at 2 to 4 hours, but by 6 hours, it's returned, right? And so this is something that we had to overcome if we wanted to have a useful molecule here of showing durable degradation of MYC. And so again, in collaboration with Tommy Moni's group, we did quite a bit of medicinal chemistry efforts on this molecule to generate this pair of molecules. It's 219A molecule that showed a bit better potency in degrading MYC, and we still showed stereoselective degradation of MYC with the less active diastereomer. This shows a high nanomolar now inhibition of MYC transcriptional activity in cells, and again, that's not observed with the less active diastereomer. It's the same mechanism of action. You have to mutate that cysteine 203 and the aspartic acid 205 to get full rescue of MYC degradation. But this molecule now is substantially more durable. And we think that this is likely due to just the improved kinetics of degradation here, that we see now durable degradation of MYC outwards of 24 hours. Now, have we improved the selectivity of this molecule? We have. When you do comparative chemoproteomics between this molecule now competing against our original probe molecule, we show nice MYC engagement and with far fewer off targets, although this molecule still has a number of off targets. We've also made an alkyne functionalized probe out of this, and we still see robust MYC engagement, again, with a number of off targets here, but far fewer than our original hit molecule. And again, when you compare it to the less active diastereomer alkyne functionalized probe, we see stereoselective engagement, again, with far fewer off targets. And when we look at now global proteomics, this molecule is exceptionally selective at degrading MYC. It's really one of the only molecules that we see going down in levels by global proteomics experiments. And so this has given us confidence that we can actually target intrinsically disordered regions and proteins, in this case, in a stereoselective manner, really strongly suggesting that there must be transient ensembles of order being formed here that we can stereoselectively recognize, covalently engage, and in this case, destabilize and degrade MYC in a relatively selective manner. And in fact, we've now been able to do this repeatedly over and over again. We've been able to covalently target an intrinsically disordered cysteine within beta-catenin to destabilize and degrade it. In collaboration recently with Amgen and Ryan Potts' team, we were able to discover a covalent destabilizing degrader. This was led by work by a postdoc in our lab, Charlotte Jamit, a senior scientist in our lab, Charlotte Jamit. This is yet again targeting another intrinsically disordered cysteine 125 within AR and ARB7 to destabilize and degrade it. Thing the Kong in our lab, postdoc in our lab, was able to recently show we could also covalently target an intrinsically disordered cysteine within IRF5 and IRF8 to destabilize and degrade it as well in collaboration with Octant. And so we think that this is a potential vulnerability, again, that we can exploit to be able to directly target these undruggable transcription factors and also be able to degrade them. But in the last few minutes here, I also wanted to touch on this bottleneck that in targeting transcription factors that I mentioned earlier, that they tend to have very fast turnover rates, right? Again, so they're being constantly kind of synthesized and degraded. And so, not only are they difficult to target, degradation may not necessarily be the best modality to tackle this class of proteins. A potential better way to tackle these proteins would be to potentially epigenetically and more durably silence the transcriptional programming of these undruggable transcription factors in diseases like cancer. Now, there are currently ways to modulate epigenetics, right? So, for example, there are a lot of histone acetyltransferase inhibitors that are being developed, like CBP and EP300 inhibitors that are being developed. Or, for example, bromodomain inhibitors like SMARCA2, SMARCA4 inhibitors. But because they're manipulating the transcriptional programming of many transcription factors, they tend to have limited therapeutic window and also dose-limiting toxicity. Now, there are also epigenome editing approaches that have been pioneered, where you can take, for example, dCas9 with guide RNA fused to DNA methyltransferases to methylate specific genomic loci. But these are massive protein complexes, and so delivery is a major problem with those molecules. And so we were thinking, could we do more precise epigenetic modulation of specific transcriptional programming with a fully small molecule-based approach? And so, in general, the way that transcription works is that you have to bring co-activator complexes that can acetylate the general chromatin region of that transcriptional programming to unravel the chromatin so that you can bring in transcription factors to initiate the transcription of target genes. And the way that you suppress transcriptional programming is that you can bring in co-repressor complexes that include things like histone deacetylases to deacetylate specific chromatin region to help compact that region to then shut down the transcription. And so one of the things that we've now been able to do is to develop recruiters against co-repressor machinery and link them to transcription factor targeting ligands to now bring co-repressor complexes to specific transcriptional loci to now deacetylate and shut down transcription in a loci-specific manner with a fully small molecule-based approach. And we're calling these transcriptional regulation via active control of epigenetic reprogramming, or tracers. Now, we're not the first ones to get into this field of targeted transcriptional manipulation. We were also inspired by work coming out of Jerry Crabtree and Nathaniel Gray's lab, where they were also able to show with their TSIP approach to use small molecules to activate transcriptional programming for transcriptional repressors like BCL6 by recruiting bromodomains or CDK9 or acetyltransferases. But for our purposes, we were really interested in developing transcriptional repressors because we were really interested in trying to silence the transcription of a lot of oncogenic cancer-causing transcription factor targets. And so this was led by a really talented postdoc in our lab, Christian Steger. And he was able to actually identify a small molecule inhibitor from patent literature against one of these kind of transcriptional repression complexes, this methyl CpG binding protein domain protein 2, or MBD2. This is a protein that recruits a major co-repressor complex known as the nucleosome remodeling complex that includes proteins within its complex like histone deacetylases to deacetylate histones and shut down transcription. And this molecule, we resynthesized and we were able to recapitulate its activity of inhibiting MBD2 interactions with the methylated DNA oligonucleotides. Now, this part of the DNA recognition in our case is dispensable because we're now rediverting the co-repressor complex to specific genetic loci. And so we were pleased to see that we could recapitulate that inhibitory activity of MBD2 in recognizing the DNA. We also found that this exit vector was sufficient to still maintain inhibitory activity against MBD2. So this could be potentially used as an exit vector for making bifunctionals. We subsequently then wanted to show proof of concept of this approach, and so we went after the estrogen receptor, since there's a lot of pharmacology associated with the estrogen receptor, including, for example, estrogen receptor agonists like this estrone derivative here. And so we then showed that in ER transcriptional luciferase supporter activity assays that either the MBD2 ligand alone or the estrogen receptor ligand alone either activates ER transcriptional activity or is silent. But when we now link these molecules together with our MBD2 recruiter now, we show very, very robust transcriptional repression of ER transcriptional activity in breast cancer cells. And this molecule is already in the high nanomolar range. Now, what's really cool about this is that this transcriptional inhibition is also quite durable. You can actually treat cells with the transcriptional repressor and then wash it out, and we can still see transcriptional inhibition of ER reporter activity many, many hours out. And so that's quite interesting that we're getting this kind of durable repression of this transcriptional activity. We also further wanted to characterize this and show that this was on target. So we were able to show that we could rescue the transcriptional inhibition of ER transcription activity with excess MBD2 inhibitor, showing that we need that ternary complex formation for this to work. We also wanted to make sure that we weren't just like disrupting the ER dimerization or recruitment of other protein partners, but that we actually required the recruitment of the co-repressor complex and HDAC activity to actually enable that transcriptional inhibition. And consistent with that, you could also rescue the ER transcriptional inhibitory activity with this molecule with an HDAC inhibitor as well. And most importantly, when you knock down MBD2, you also get rescue of that transcriptional inhibitory activity, showing that this is on target. Now that's with a relatively artificial luciferase reporter assay, and so we also wanted to show that we could manipulate endogenous ER target genes, and so we were able to show that we could downregulate ER target genes like ER itself, as well as downstream targets. And again, that's rescuable with MBD2 knockdown. We also did global RNA-seq, and while we see hundreds of genes transcriptionally being altered with this molecule, when you do pathway analysis on this, it's really just the ER pathway that's being manipulated. So we're not globally screwing up transcription. We're quite selectively manipulating ER transcription, transcriptional programming in these breast cancer cells. We also wanted to look into ATAC-seq to look at chromatin accessibility. And what we were able to find through this was that we're quite selectively manipulating chromatin accessibility at target genes that have ER motifs. And so we're quite selectively just manipulating ER programming in these cells and not just kind of globally screwing up transcription. We also wanted to see whether we could do this with other recruitment of other co-repressor complexes. And so given that the epigenetic field has quite a bit of pharmacology that's out there that's quite advanced, we were also able to link a polyclonal repressor complex inhibitor, a PRC2 inhibitor, and link it onto the estrone targeting ligand, and again show that we can nicely suppress ER transcriptional activity through the recruitment of other transcriptional repressor complexes as well. So that showed proof of concept that we could do this with estrogen receptor, but we wanted to also show that we could do this for example, a target with perhaps more kind of clinically unmet need, such as, for example, the truncation variant of the androgen receptor, ARV7. Now, this is a major undruggable transcription factor that drives androgen-resistant prostate cancer cells. And the reason why it's undruggable is that ARV7, unlike full-length AR, is missing the entire ligand binding domain or steroid binding domain that testosterone binds to, as well as all the AR antagonists like the standard of care for this cancer, enzalutamide. And all that's left is an intrinsically disordered N-terminal binding domain or DNA binding domain that's now highly intrinsically disordered and now permanently activated. Now, in these cancers, the standard of care is enzalutamide, but in these cancers that express both AR and ARV7, you only get partial inhibition of total AR transcriptional activity because the other half of this is driven by ARV7. But both AR and ARV7 still bind to the same AR transcriptional loci. And so if we can epigenetically shut down the programming of AR, we'll also by default shut down the transcriptional programming of ARV7 as well. And so we wanted to test this. And so we used the selective androgen receptor modulator ligand that again only partially inhibits AR transcriptional activity in these cells because this binds still to the ligand binding domain. But when you now link this SARM ligand to our MBD2 recruiter now, we get over 90% suppression of AR transcriptional activity in androgen-independent prostate cancer cells bearing both AR and ARV7. And you can rescue that with an HDAC inhibitor, just like with the ER tracer. We see downregulation of AR target genes by qPCR. And again, if you knock down MBD2, you get rescue of that transcriptional inhibitory activity. And when we do RNA-seq, we see global repression of all the AR, ARV7 target genes. And again, when you do pathway analysis on this, we get enrichment of AR response genes showing up. And so we're not just globally screwing up transcription, we're relatively selectively targeting AR transcription. And so we're quite excited about this approach of being able to now use small molecules to essentially do epigenome editing at specific genetic loci now by now driving co-repressor complexes to specific transcription factor start sites to now kind of durably silence transcriptional programming of specific transcription factors. And so I'll end there. We're quite excited about this approach and hoping to expand this approach. And this is just really the tip of the iceberg of what we're working on and using covalent chemoproteomic approaches to tackle undruggable targets, to expand on targeted protein degradation technologies and go beyond degradation, like we've shown, for example, with targeted transcriptional modulation with tracers. And with that, I'll thank my lab at UC Berkeley. And also many of the stories that I talked about today, I think actually all of the stories that I talked about today were in collaboration with Novartis through our Novartis Berkeley Translational Chemical Biology Institute. And also thank you to our other funding sources, including the Mark Foundation for Cancer Research and also NIH. And happy to take questions in the remaining time.
Amazing. Thank you so much, Dan. That was super inspiring. I mean, really seeing these kind of unusual warheads, that people don't use every day to drug transcription factors like MIG is really extremely impressive. So yeah, thank you again
Thanks.
for showing your work. We have a couple of questions in the audience already, and I'll just go through them. So there's a question from Cynthia Berry. For your MYC ligands, how much of the activity would you contribute to the covalent warhead versus the non-covalent interaction with the protein? That I think was for the sulfonyl aziridines that you showed.
Right. So in our paper, we actually were able to generate the unreactive versions of those molecules, and they completely lose activity. And so, at this stage, most of the activity is still driven through the kind of K-enact, right? But still in a stereoselective manner, right? So, yeah, this is something that we're further optimizing with further medicinal chemistry efforts to hopefully be able to achieve better Ki over K-enact.
Awesome. Yeah, wonderful. And then with respect to kind of the cysteine, so Neil Bola has a question. Have you seen only targetable cysteines in the disordered regions of transcription factors, or is there a possibility to potentially have targeted lysines in that
Oh yeah,
area?
I mean, it's not nearly as big as our kind of cysteine database, but we've certainly identified intrinsically disordered and what appear to be aberrantly active, for example, lysines within disordered regions of proteins, including transcription factors. I think with cysteines, it's just we have a lot more chemical matter and libraries to target them, but I'm sure this is achievable with other amino acids as well.
Awesome. Yeah, and I think you elaborated on this on the earlier question already, but Alex Garasudo has a question. Oh, sorry, it's just skipped because there came a new question. Is it possible to convert the covalent probes targeting novel E3 ligases to non-covalent binders? He's curious if covalent degraders maintain catalytic mechanism of action.
Right. So I think with enough medicinal chemistry, if you're able to achieve enough Ki against your E3 ligase, you can hopefully eventually take off the irreversible warhead, or you can convert it into a covalent reversible molecule. In terms of catalytic activity, so if your covalency is on the side that you're recruiting, like an E3 or a D-ubiquitinase or whatever, you're still going to maintain that catalytic activity. And you've basically turned your ternary complex into a binary complex where there's actually advantages with that. You could really now further uncouple PK and PD, right? Because now, even if your drug washes out, your covalent protac that's bound to your E3 will continue to engage your target until the E3 turns over, right? And so there's certainly advantages with that, and you can still maintain catalytic degradation activity. Now, if the covalency is on the target side, obviously that's going to be stoichiometric and not catalytic anymore. And so you have to think about that.
Yeah, that totally makes sense. And then, yeah, there's a question from Yoshi Ishihara, basically about the high turnover rate of MYC. So for those rapid turnover proteins, is it a matter of metchem optimization to improve potency and PK to maintain sufficient compound concentration at the target environment, or is the protein turnover rate inherently a no-go for a degrader approach?
I don't think it's a no-go for degrader approaches. I mean, certainly there are protacs being made against these high value, these high turnover targets that
Yeah,
are clearly effective. And so, it's definitely not a no-go for sure, but it's just a challenge that you have to overcome, right? So, you either have to improve your PK of your molecule, so you're getting sustained engagement, and or yeah, it's just a challenge if you're going to have your molecule there constantly to be engaging the new targets that are being synthesized. And again, that's why we kind of came up with this tracer approach, right, to hopefully get more durable
yeah,
silencing of those transcriptional loci. Yeah.
awesome. Yeah, and in the similar vein for the MYC inhibitors, what is the half-life in a GSH assay or a similar intrinsic reactivity assay for those kind of novel warheads?
Right. Yeah. So the sulfonyl aziridines that we generated are on the order of like an hour or two. So they're on par with like currently clinically approved covalent drugs that also have somewhere between 40 minutes to an hour GSH half-life. I would say though that some of the newer warheads that we're developing that hopefully we'll be able to show in the future are now having like over 12-hour GSH half-life, right? So, we're
Mhm.
really looking into like really, really tempering back the reactivity of these warheads while also incorporating in stereochemistry and more drug-like properties and such.
Amazing. Amazing. Yeah. What would you say, another question, what would you say are the strategies that enable rational design of probes for intrinsically disordered proteins in general, with that they're, have dynamic and flexible structures?
Oh, yeah. So, for storytelling purposes, I gave this really beautiful story of saying that we rationally designed for the bespoke purpose of targeting IDR regions. In reality, right, we were generating these libraries that have a higher sp3 content, stereochemistry, tempered reactivity, and then we applied it to MYC, and it fortuitously worked. But I would say that, so I don't know about rational design strategies for specifically targeting IDR regions, but as we've, right, and this is something that many other labs have shown too, whether it's Stu Schreiber's lab and Ben Kravat's lab and others, but I think the higher degree of three-dimensionality you have in your molecules and the ability to be able to create these kind of stereochemically paired molecules, I think has given us larger access into a lot of these very challenging targets that we haven't been able to previously access with kind of your traditional covalent libraries that might be commercially available. And so our hit rate has gone up in targeting a lot of these much more challenging molecules. And again, with the stereoselectivity that you observe there, it just gives you much more confidence that these molecules are hitting, something that will hopefully be progressible, right?
Amazing. Yeah, and we have a question from Gufang, something that Dan and I discussed actually before the meeting. So thanks for a fantastic talk. Could you comment on the tracer versus RipTag approach and tracer versus TSIP approach also? Like what are, the design utilities and the advantages of each?
Yeah, so I mean, so far, Nathaniel Gray and Jerry Crabtree with their TSIP approach have really focused on transcriptional activation and incredibly beautiful work there. And their main application so far has been against activating BCL6 repressive loci to induce cell death of cancer cells. And so, this is very analogous to that. It's just repression, right? And at least in my head, a lot of the targets that I've really cared about going after for transcription factors have been oncogenic, not tumor suppressive transcription factors, and so we'd want to shut those programmings down as opposed to activate them, right? And so I would say that they're very analogous. They're just opposite. Instead of activation, we're just repressing. And I think the question in the future will be, can we start to rewire transcriptional programming with these kinds of approaches as well, right? In answer to the RipTac question, so I did show that we're not necessarily just like disrupting like a native protein complex or something. It's requiring the recruitment of the HDAC activity to the specific loci because we can rescue with HDAC inhibitors, right? So this does seem to be kind of more like a targeted transcriptional modulation rather than like sequestering a target or whatever, like with the RipTac approach. Although I have to conjecture, their RipTac molecule that they have going in is like a BRD4 inhibitor linked to an AR antagonist, right? So those are transcription factor linked to an epigenetic modulator. So I wonder if their molecule is also acting through kind of some kind of transcriptional DNA trapping or transcriptional modulation of AR in addition to their kind of reported mode of action. It's an interesting question worth pursuing.
Yeah, certainly something that we might need more data on, right?
Yeah.
Amazing, amazing. Yeah, maybe in the interest of time, we have so many questions, but maybe one
Sure.
more, and then we'll close it up. Eva is wondering, how did you identify the most tractable residues from your database, and how did you prioritize those residues to go after?
Yeah, so our ligandability database is a little bit different from like many of the ones that have been published out there where they're using a scout ligand in competition with iodosetamide. What we've done is taken all of the chemoproteomic pro-modified site data that we have and just aggregated it. So it's like thousands and thousands of data sets. And when you look up a protein in there, maybe like a particular protein will have like 7 modified cysteines on it, but one of those modified cysteines shows up disproportionately in terms of being modified. And that's in 99% of situations correlated with the reactivity of that cysteine, and also when you do a covalent screen against it, with the ligandability of that site. And so we've used it as a prioritization metric for which cysteine within a particular protein is the most reactive and most likely to be ligandable. And if you look at all these kind of aberrantly reactive sites, they're of course highly enriched in all the catalytic cysteines and previous sites like allosteric cryptic sites that have been interrogated already previously. And so besides the previously annotated sites, there's like tens of thousands of unannotated sites that appear to somehow be aberrantly reactive, right? So that's kind of how we assembled that database.
That makes a lot of sense. Awesome. Dan, thank you again for a super inspiring and motivating talk. I think you showed that problems that have traditionally been very challenging to crack, we have now a path to solving them. So I really am super fascinated by your work. I want to thank all the audience for the engagement here and the questions. We had many more. Apologies if we couldn't answer all of them in time. So let's give a huge round of applause to Dan again, virtually.
Thank you everyone for showing up.
Exactly. Thanks everyone for coming. And we will be here next week and we will talk about patenting strategies for small molecules with Lucas Watkins and Alex Burney. So that's going to be an interesting topic if you're looking to get into the IP landscape. So completely different topic, but encourage everybody to dial in today. So thanks again, Dan, for an amazing talk, and we will see you then.
Great. Thanks.
Bye-bye.